Good morning, dear students of class 12 science. Welcome to biology class. We are dealing with the chapter biotechnology and its application. So in the last class, we have learned about the use of biotechnology in the field of medicine or in the field of healthcare. So today, we are going to learn about the stem cell technology. Okay. So stem cell technology is mainly a multidisciplinary branch. It's got a huge branch, okay, that mainly contributed by the cell biologist, those who study cells. Then we have got genetics, those who study the genes. Then we have got embryologists, those who study the embryos. Then we have got clinicians for the clinical applications of manipulated stem cells. And this stem cell technology is mainly a tool for the discovery of drugs, for the drug development and the therapeutic measures for controlling mainly, let's say, for cancerous and then non-cancer diseases. Cancerous means those cells which are not behaving in a normal manner, okay, they don't follow the normal cell cycles. And non-cancerous means those who used to go through a normal phase of cell cycles and cell divisions. So in this type of diseases we can go for stem cell technology. So actually, what do you mean by stem cells? Since we are going through the stem cell technology, so what do you mean by stem cells? So stem cells are mainly the undifferentiated, which are not differentiated into any types of tissues or organs. Clear? So they are undifferentiated cells. They are unspecialized cells, and they are totipotent cells. They can form any types of tissues, any type of cells or organs in our body. Clear? So stem cells, they are undifferentiated cells, they are unspecialized, and they are totipotent cells that can divide indefinitely for self-renewal. Okay, and they have the potential to differentiate into different types of specialized cells. This is one of the most important features of stem cells, that they have the potential to differentiate to any type of specialized cells. That's why we are using these cells in this stem cell technology for the treatment of different types of diseases. So let's talk about the characteristics of the stem cells. So stem cells, they are the progenitor cells. They will give rise to different types of cells in our body, and they are capable of self-renewal. That is, they can multiply by mitosis to replenish their own population. Mitosis means the process of cell divisions, where there will be a constant number of chromosomes between the parental cell and the daughter cells, and by the help of this mitosis cell division, the stem cells, they can undergo self-renewal and they can multiply in number. Number 3, the stem cells have the capacity of multi-lineage differentiation. They can differentiate it into any type of specialized cells. Clear? Number 4, stem cells have the potential for treatment of malignant and non-malignant diseases. Malignant disease means those diseases which can spread all over the body, like for example cancerous, malignant tumor, which can spread throughout the body, okay, by the process called metastasis. And non-malignant disease means which are confined to a particular location, particular positions. Number 5, gene transfer into hematopoietic stem cells can help the treatment of many genetic or acquired diseases. Genetic disease means which are being inherited from the parents to the offspring. So those diseases can also be treated by the help of stem cell technology. Next character is neuronal stem cells. It can be used for the neuron replacement in Parkinson's and Huntington disease, which are the nervous disorders. So such disease can also be treated by the help of the neuronal stem cells. These are the characteristics of the stem cells. Now let's talk about the potency of the stem cells. Okay, so potency is the capacity of undifferentiated cells to differentiate into specialized cell types, because we are saying that stem cells have the capability to differentiate into any type of specialized cells. So over here the term potency means the capacity of those undifferentiated cells which has not been differentiated till now into any cells or any tissues. So those undifferentiated cells, they will differentiate it into specialized cell types, and that particular term is called potency. So therefore stem cells, they may be totipotent, they may be pluripotent, they may be multipotent, oligopotent, or they may be unipotent progenitor cells. Clear? So we'll go through one by one what the totipotent means. So totipotent stem cells are those cells that can differentiate into embryonic cell types. These cells are produced by early blastomeres. It's formed by the first divisions of the fertilized egg and by the cells of the morula. So each totipotent cells can develop into a complete organism. Already in the chapter of fertilization of human being, we have learnt about these terms like what is morula. Okay, we have learnt about the embryonic cells, all those things. So totipotent cells can differentiate into embryonic cell types. Number 2 is pluripotent stem cells, which are the descendant of the totipotent cells and can differentiate into a cell types. But the cells of the inner mass, the cells of the inner cell mass and the 3 embryonic germ layers are pluripotent. If you remember the structure of trophoblast, we have the inner cell mass and the 3 embryonic germ layer means ectoderm, endoderm and the mesoderm, which will give rise to any type of cells or tissue in our body. Okay, so pluripotent stem cells are the descendant of the totipotent cells and they can differentiate into all the cell types. So the cells of our inner cell mass and the 3 embryonic germ layers, the ectoderm, endoderm and the mesoderm, they are the examples of pluripotent stem cells. Then we have got multipotent stem cells which can differentiate into a number of cell types, but only those of closely related family of cells. Then we have got oligopotent stem cells which can differentiate it only into a few cell types such as lymphoid or the myeloid stem cells. Then we have got unipotent stem cells which can produce only one cell type by their own type. Uni means one, so they can only produce their own cell type, but they have the property of self-renewal such as progenitor cells and the muscle stem cells. Okay, so these are the potency of the stem cells. So we have got the totipotent stem cells, we have got the pluripotent stem cells, we have got the multipotent stem cells and oligopotent stem cells, then unipotent stem cells. Now let's talk about the types of stem cells. Okay, just listen very carefully, the types of stem cells. So based on the source of the origin, the mammalian stem cells have been mainly classified into 3 types. Those are the embryonic stem cells, the fetal stem cells and the adult stem cells. Okay, these are the 3 types of stem cells which are present in case of mammals based on their origin. So first one is embryonic, second one is fetal stem cells and third one is adult stem cells. Clear? So embryonic stem cells, they are mainly totipotent cells. We already mentioned that embryonic cell types, they are totipotent stem cells which are isolated from the embryo at the early stage of blastula or inner cell mass of blastocyst. Clear? Then we have got fetal stem cells. After 8 weeks of development, the embryo is called the fetus, that we have already done. Okay, so the primitive stem cells located in the organ of the fetus are called the fetal stem cells. Clear? Then we have the third type which is called the adult stem cells, which are also called the somatic stem cells, and these are mainly used to maintain and repair the tissues in which they are found, and mainly they are found in children as well as adult but are small in number. For example, we can say the bone marrow cells. So the bone marrow cells is a rich source of adult stem cells. Okay, so these are the 3 types of stem cells which are mainly found in case of mammals based on their origin. So first one was embryonic stem cells, number 2 will come to the fetal stem cells, and number 3 is the adult stem cells. Okay, now let us see the few diseases where the stem cells can be used. Okay, those diseases where we can mainly use the stem cell technology for the treatment. So it can be used for diabetes also, it can be used for rheumatoid arthritis, the joint pains. Okay, it can be used for Parkinson's disease, it can be used for Alzheimer's disease, it can be used for osteoarthritis. Then we can use the stem cell technology for the treatment of stroke and traumatic brain injury diseases. It can be used for the learning disability due to the congenital disorder. It can be used for spinal cord injury repair, okay, heart infections, anti-cancer treatment, baldness reversals, missing teeth treatment, repair hearing, restore vision, amyotrophic lateral sclerosis, Crohn's disease and the wound healing. So these are the few diseases where we can use the stem cell technology, okay, and we can mainly cure those diseases. But what are the disadvantages of this stem cell technology? Okay, what are the drawbacks? So stem cell treatment, they require immunosuppression. Immunosuppression means we have to suppress, we have to make the immune system weak. Clear? Because we have already learned about immunology also, okay, where our immune system is to work against the foreign cells. Clear? Where they used to recognize the self cell and the foreign cells. So this stem cell technology, they used to work on the property of immunosuppression, where we have to decrease the property or the strength of the immune system, because the requirement of radiation before the transplant to remove the patient's previous cells. So we are going for radiations where we have to remove the patient's previous cells, and the patient immune system may target the stem cells. So that's why we have to make the immune system very, very weak, so that they will not be able to target the stem cells. So the use of the stem cells from the same patient who is being treated is the solution of this problem. If you are going to use the same stem cells of the same patient, then there is no need to go through the immunosuppression. But whenever we are using the stem cells from the other donor cells, in that case that cell will be the foreign cells, and whenever the foreign cells get interest within our body, our immune system will work against it. Okay, so the only solution to this problem is that we can use the stem cells from the same patient, and in that case there is also no need to go through the immunosuppression. So this is the few disadvantage, or we can say the drawbacks of the stem cell technology. So today we have just finished about the stem cell technology. In the next class we will learn about the application of biotechnology in the field of agriculture. So this much for today's class. So we will continue with the same chapter in the next class. Thank you.